the little vessels, if we take a cross-section of those vessels, this is what it looks like. So these little circles are the red blood cells. Here's the wall of the vessel. And then in this sort of lighter shaded pink here is the smooth muscle cell wall, okay? It's the wall of the vessel. And the smooth muscle cells are very important for keeping the vessel at the right tension. So when you get high blood pressure, your cerebral blood vessels relax because it wants to make sure that it's appropriately responding to the change in pressure, and the smooth muscle cells are critical to that response to the change in blood pressure. And the smooth muscle cells are sitting here. These are in blue, so they are surrounding the vessel, and the key protein we're going to talk about today is Notch3, and Notch3 is sitting there at the membrane of this smooth muscle cell, okay? So it turns out that mutations, so changes that are inherited in the Notch3 gene, is what causes CADASIL. So why is that? We don't know. Let me just stick with that. These are supposed to come in order so that it didn't get too busy, but it's too busy. So we'll just go, let me just take you through. So this is the Notch3 protein. It's sitting here on the membrane of the smooth muscle cell within the vessel wall. And it's a big protein. It interacts with its environment. When there are changes that are associated with CADASIL, what happens is the pattern of the little protein components change. There should be an even number of one of these protein components called cysteine. When there's a mutation that causes CADASIL, you get an odd number. That's just one mechanism by which Notch mutations affect the protein. So it affects the structure of the protein. Now normally, when Notch3 is sitting there, it functions to take signals from the external environment and transduce it to the intracellular space. So the vascular smooth muscle cell can then respond to clues from the outside. And this protein is critical to doing that. So it gets a signal from the environment. And part of that signaling is that it gets cleaved by an enzyme right at the junction of the membrane. And then this intracellular part now moves through the cell, and now that it's moving, it can then initiate signaling to start other functions going on in the cell, okay? So that's one important part of Notch3. So one problem could be that when you have mutations in this Notch3 protein, you get a change in its normal function. There's something wrong with the way it responds to its environment. There's something wrong in which it lays itself ready to be cleaved, and you can impair the function of Notch3. So one possibility is CADASIL is caused by a lack of normal function of Notch3 because of mutation. But another thing that we've learned, and I'm going to show you a little bit of pathology in a few seconds, is that when we look at the skin, if we look at vessel pathology from patients that have CADASIL, there's abnormalities in the vessel wall. There's also accumulation of protein. Very odd, unusual, and shouldn't be there kind of protein collections. And one of the peptides that are near those protein collections is Notch3. So when this protein gets cleaved, it releases this intracellular part, but it also releases this extracellular part. And this kind of goes floating off. But when there is a patient with CADASIL, you get an abnormal accumulation of this protein, which then sits there. And another hypothesis is that too much accumulation of this Notch3 protein also causes damage to the cells in the vessel.